Hello students, welcome to another lecture of human anatomy and physiology, and in today's lecture we will be studying about cell division. So let's start. Now most damaged cells basically they can be replaced by growth and division of other similar cells. Now the frequency with which cells can divide depends upon the type of tissue. So cell division is basically a carefully regulated process to allow effective maintenance and repair of the body's tissue, right? So cell division can be divided into two types, first being mitosis and the second being meiosis, right? So cells with nuclei having 46 chromosomes, they can divide by mitosis, and mitosis is a process that results in the generation of two new genetically identical daughter cells, right? The only exception to this is the formation of gametes. Gametes are basically the sex cells, right? That is the ova in females and spermatozoa in males, and they divide by meiosis. Now a cell cycle. So cell cycle is basically a period between two cell divisions, right? And this particular cell cycle has two phases. One being the M phase, which is also known as mitosis, and the other is interphase. We'll study about these two phases in our coming slides in details. Now this is a diagram of the entire cell division. You can see there are two phases, one being your M phase where the cell divides and the other being your interphase. Interphase is further subdivided into three categories, and interphase is mainly a phase of cell growth and M phase is the phase of cell division. Now this is a basic diagram of mitosis where a cell starts dividing and the ultimate result is the formation of two identical daughter cells. This is basically a continuous process involving distinct stages. The first being prophase, the second being metaphase, third being anaphase, fourth being telophase, and the last being cytokinesis, right? So we'll be studying about all these phases in details, starting with prophase, right? So during this phase, what happens? The replicated chromatid, they become tightly coiled with each other. Each of the original 46 chromosomes, which are known as chromatid at this stage, they are paired with its copy in a double chromosome unit. The two chromatids, they are joined at the center by centromere. In this particular state, the mitotic aperture, it starts appearing, which consists of two centrioles which are separated by mitotic spindle, which is formed from the microtubule. And the centrioles migrate one to each end of the cell, and the nuclear membrane or the nuclear envelope basically, it starts disappearing. This is the basic structure of your prophase. What happens? The nuclear envelope starts disappearing. You can see the formation of mitotic spindle, which helps in the basically the separation of the centriole to two opposite poles. Centromere is the area where the chromosomes are joined together at the center, and chromatid are basically the replicated chromosomes. They join together with the help of the centromere. Next phase is your metaphase. So metaphase is basically a phase where the chromatid, they align on the center of the spindle attached by their centromere, right? And mitotic spindle is basically a imaginary line. As you can see in this particular slide, the chromatids have aligned on the center of the spindle attached with the help of their centromere. Now after all the chromosomes are aligned on the metaphase plate, each pair of the sister chromatids, they split at the centromere, separates and move along the shortening spindle fiber to opposite side of the cell. Now the number of centromere and chromosomes within the cell is doubled. Because why? Because they are separated, right? The separated chromosomes have only one chromatid at this particular stage. As you can see in this particular slide, sister chromatids have formed and they have moved to two opposite poles. Now coming to the telophase. So chromosomes each with one chromatid arrives at opposite pole of the cell, and a new nuclear membrane starts forming around each of the two daughter nuclei which are identical to each other. the spindle fibers begins to disperse and the chromosomes, they decondense. Now chromosomes, they are no longer visible under the light microscope, right? Now here what happens, the number of centromere and the number of chromosomes within each of the two identical daughter cells is similar to that of the parent cell before it underwent mitosis. Now here you can see the nuclear membrane has started, appearing, and the spindle fiber or the mitotic spindle has disappeared. Now the last stage is your cytokinesis. In cytokinesis, the nucleus, although the nucleus divides during mitosis, the cytoplasm does not, right? Following mitosis, cytokinesis is the process where the cytoplasm of this cell is divided to form two identical daughter cells. Right? In animal cell, the center of the cell contracts, pinching the cell into two daughter cells, each of which contain a nucleus with a complete genome. So this is a process of cytokinesis wherein the cell is being pinched from the center and there is formation of two identical daughter cells, each having its own genome. And the next part is your interphase. In interphase, the cell does not divide, but it prepares itself for division. It is basically a growth phase. This is the longer phase as compared to mitosis, and it is subdivided into three stages. First being the first gap phase, which is G1. Second being your synthesis of DNA, that is S phase. And the third being your second gap phase, which is G2. So in G1, what happens? The cell grows in size and volume. This is usually the longest phase and the most variable in length, right? In the second phase, which is the synthesis of DNA, the chromosome replicate, forming two identical copies of DNA. Therefore, following the S phase, the cell now has 92 chromosomes, right? And the third is your second gap phase, which is your G2 phase. In this particular stage or phase, there is further growth and preparations for cell division. So thank you so much, dear students. This was all in today's lecture. Today we studied about cell division. If you have any queries, any doubt in today's lecture, please feel free to reach out to me. Thank you.